Okay, so I can only video for like 15 minutes at a time on this, but we'll give it our best shot. So what I thought we would just do tonight is basically just kind of work our way through some of the objectives for the different chapters that are represented on the test. And I would love to start with whatever material you guys feel like you would like the most review on. We can always come back to other things as we have time, but if you have very specific topics that you'd prefer to start with, or a chapter that you thought was more difficult, like let's start with that.
I don't have a specific one.
I don't either.
Guess just
like the genetics one?
genetics. Okay, yeah, we can start there. So let me just move over here to that's cellular adaptation. Okay, so there we go. So chapters 4 and 5 are the genetics chapters. I would definitely recommend like studying them together, you know, because basically in chapter 4 you kind of have like a review of the basic normal kind of information. And then chapter 5 just talks a little bit more about some of the disorders. But the reality is chapter 4 talks about, you know, the Mendelian principles and doing a Punnett square. And then you just learn about doing some of the examples for specific disorders in chapter 5. So those two chapters really work kind of hand in hand. So some of the first couple of objectives here, and please interrupt at any point if you have some specific questions. This is not meant to be lecture by any stretch of the imagination. I just want to make good use of your time, but interrupt at any point if you want to go through something different. In terms of the structure and function of DNA, you know, hopefully that was mostly review. That's not really the stuff that I focused on in terms of the exam because that's more general bio stuff. DNA mutations, I mean, that really even goes back to the chapter about cellular adaptation because we talked about things that can cause cellular injury. One of the ways cells can be injured is obviously by having something cause a mutation to DNA. So we're thinking about things like radiation and those kinds of things that can actually directly cause some DNA mutations, or we can have, you know, problems with chromosome number where we end up with one too many or one too few, which are some of the disorders that we look at a little bit in chapter 5. Which mutations can be inherited? So we all kind of end up with mutations that are happening all the time. But which are the mutations that we can actually pass on to our offspring? So if I have a mutation in one of my general somatic body cells, right, that's not a cell that's going to go to my offspring. So where would the mutation have to be if it was going to be something that was going to be passed on? Don't make it too hard.
The dad?
It's got to be the sperm or the egg, right?
Yeah.
So if you're going to... So if you're going to pass that mutation on, it's got to be something that's in one of those germ cells. So some of the other mutations, it might cause harm to us, but it's not going to be something that can be passed on. It's only going to be a mutation in the germ cells or the sperm or the egg that can actually be inherited and passed on. So that's kind of an important piece. All right, so the next one is to talk about the different types of RNA, and that kind of feeds into transcription and translation. There definitely are a couple of questions on the exam about those particular pieces. So it's definitely one piece to look over, see if I can kind of see where we start here. So your 3 types of RNA are right here. And essentially, as we're looking at the 3 types of RNA, this is going to describe the process of transcription and translation. So we're really talking about how do we get the directions from the DNA that never leaves the nucleus, right? That DNA is a very stable molecule. It always stays in the nucleus and it contains that entire genetic code. So how do we get that message then from the DNA inside the nucleus out to the cytoplasm where the protein synthesis is actually happening on the ribosomes? And so that's the process of transcription and translation. The first part of it is transcription. And I just always remind people, you know, think about a transcriptionist in a courtroom, right? Their job is to essentially make a copy of everything that happened in terms of those court proceedings. So they're recording everything. And that's really the process of transcription. When we think about protein synthesis too, it's making a copy. So messenger RNA is really the piece that's responsible for that. So messenger RNA, the DNA unzips, and that messenger RNA comes and makes that complementary copy for just the protein that it's coding for. So, even though we have all the genetic material for every protein in our body in the DNA, each cell just knows which portion is appropriate for it to use. So that messenger RNA will make a copy of the genetic material for one specific protein, and then it leaves the nucleus. So the DNA stays in the nucleus, but the messenger RNA leaves. So it carries the message, right? Name messenger RNA. It carries the message out then to the ribosome. And part of the ribosome is literally this ribosomal RNA. So you can just kind of think about those things together. Ribosomal RNA is literally part of the ribosome where protein synthesis is actually taking place. So now we've taken care of two types of RNA. We've talked about messenger RNA and ribosomal RNA. The last piece is transfer RNA, or sometimes referred to as tRNA, and it's transferring the different amino acids to that ribosome for assembly. And that's the piece that's referred to as translation. It's basically translating those directions that came from the nucleus now into a brand new protein. So again, transcription is making the copy. So messenger RNA is really responsible for that. Messenger RNA goes into the nucleus, makes the copy of the portion of DNA that it needs, right? And then it takes it out from the nucleus and it goes out to the ribosome where this protein synthesis is going to actually take place. And then the translation piece is essentially once that messenger RNA comes together, it comes into contact with the ribosome, and these different little tRNAs, transfer RNAs, are bringing amino acids to the ribosome for assembly. So this is a picture of transcription. So here we are inside the nucleus. We've got our DNA that's kind of unzipped. And here's my mRNA, or my messenger RNA, that's making that complementary copy. You can see how those base pairs line up there. So that's the copying piece. Then that messenger RNA is going to detach from the DNA. It's going to leave the nucleus. And you'll see here, this is the ribosome. Okay, so I've got my messenger RNA now that's come. It's joined the ribosome, which has ribosomal RNA in it. And then here are the tRNAs. And so you'll see this transfer RNA has what they call the anticodon, right? Because it lines up to the 3 base pairs here. And each one of them has this little amino acid that's attached to it. And so it starts making this really long chain of amino acids that are coming together now and forming a protein. And then the last step is that the cell has to actually take that protein and form it into the right 3-dimensional shape. You can literally have the right order of amino acids and actually still have the protein be dysfunctional because it's not in the right shape. So it has to be folded into exactly the right shape for it to work correctly. So that's really the process of transcription and translation. And there will be a question, there are questions on the exam asking you to distinguish between transcription and translation. And there are questions on the test that make you know the different types of RNA. You have to know mRNA, or messenger RNA, ribosomal RNA, and transfer RNA, and what role each of them play in protein synthesis. This little video, I like visuals, so I won't show it now in the interest of time, but this is a great little video if you just want to kind of put it together in a slightly different way. That is really a nice picture of what protein synthesis is kind of looking like. Okay, so that's where some of those questions, gosh, that's probably 3 questions from that particular chapter right there. So, kind of helps you know where to focus your studying on that piece of it. Just remember your autosomes are your 22 pairs of normal body chromosomes, right? Not your sex chromosomes. So your autosomes are those 22 pairs. So we're going to talk more about them when we talk about autosomal dominant and autosomal recessive. That's the key piece there. When we're talking about those, we're not talking about an X-linked disorder. We're just talking about regular autosomal disorders there. Chromatin is just the stuff that makes up the chromosomes. That's enough to know about that, but it's the material that's making up those chromosomes. The key thing about mitosis versus meiosis, you don't need to have the stages of mitosis memorized or anything like that. You probably did have that memorized at some point for biology or whatever. That's not really what the point of it is. The key point in this chapter is just to take away from the fact that recognizing that meiosis is happening only again in the germ cells. because we only want 23 chromosomes, not 23 pairs of chromosomes there. So that when we have the sperm and the egg and they come together, we end up with 46. So meiosis basically goes through a second level of division. Mitosis, we end up with two daughter cells that look exactly like the parent cells. The chromosomes have duplicated, they look exactly the same. In meiosis, we're literally cutting the number of chromosomes by half. So it basically goes through a second division, and we only see that happening then in the germ cells. Karyotype is just a picture, so literally they can take cells that are, they usually, I think, use lymphocytes, and they arrest them in a specific phase of mitosis where they can really see all of the separate chromosomes, and then they line them up. So this is how they diagnose a bunch of different types of chromosomal disorders. I know when I was in high school biology, we actually got to make our own karyotype. That was a pretty cool project. So you could line up your own chromosomes and find the X and the two X's, or the X and the Y, depending upon, and that was actually a pretty cool thing. So you can actually see the different chromosomes when you're going through that process. They can use that for diagnosing disorders. They can even now use that to diagnose a lot of disorders prenatally, which, you know, prenatal diagnosis is kind of in that chapter 5. So the Barr body principle is right here. Don't overcomplicate it, but there is one question on the test about the Barr body principle. So essentially what we're just saying is, interestingly enough, when we're dealing, so a normal male is going to have XY for those chromosomes, for the sex chromosomes. A female is XX. Well, the reality is we actually only need one X, right? That one X is going to have all the genetic material that we need. And so essentially one X will be expressed. The other one is just going to be inactive. So we kind of just say it's like an inactive mass of chromatin. So the material here that makes up chromosomes. So we've got one active X and one inactive that we call the Barr body. So why that's important is that Barr bodies actually show up, they'll stain differently if we're looking at it under the microscope. And so with a trained eye, you can actually identify that Barr body. And so when researchers are looking at that and trying to maybe diagnose some of those different types of disorders, they basically know that the number of Barr bodies that they see plus 1 is the number of X chromosomes. Okay, so let's think about that for just a second. So a normal female then, who's XX, how many Barr bodies would they see when they looked at it under a microscope? One, absolutely. For a normal male who's XY, how many Barr bodies should there be?
1, 2.
Remember, it's the number of Barr bodies plus 1 equals my number of X chromosomes. So actually it's 0, right? We wouldn't have any in a male because for the male, that X, they only have one X, so it's always going to be expressed. Versus the female that has 2, so we've got one Barr body plus 1 active X. Does that make sense?
Yeah.
So the male would have 0 Barr bodies. Females should have 1. So where that gets to be interesting is if we look at some of the problems in chapter 5, right, that deal with chromosome number. So if you think about that, in that instance where we're talking about Turner syndrome, where we have a phenotypic female, but if we were to look at the chromosomes, it's actually XO. In other words, they only have one X. They're missing that second X chromosome. So that is actually a female, but they wouldn't actually have any Barr bodies. Their number of Barr bodies would be 0. Atypical for a female, and that would be our first clue that there's probably a genetic disorder going on. Same thing for Klinefelters, which is a phenotypic male, but they're actually XXY, so they actually ended up with an extra X, even though they're a male. So although males should normally have 0 Barr bodies, in Klinefelters, they would actually see 1 Barr body when they looked under the microscope and be able to identify that they have a genetic disorder going on. So that's what the Barr body principle means. It just means if there's an extra X, the one is inactive. It's not actually being expressed. And interestingly, in your normal healthy female, so about half of the X's that get expressed will be from the mom, and about half of the X's that get expressed will be from the dad. So there's where we get a lot of our genetic variability even between siblings. So kind of an interesting thing to think about there. So we've kind of been alluding to a couple different terms. Genotype. Genotype just means if we're making that Punnett square, genotype is big B, little b, or big B, or little b, little b. So just, what are those alleles? Alleles just means one of those two letters. One of the